here as a clinician, so I know most of you out here are clinicians, and so we're hearing so much about like advances kind of at the micro level. So I'm going to take it kind of back up to macro level, nutrition, whole foods nutrition, and the intersection between epigenetics and inflammatory responses. So today I'm going to talk about oxidative stress. We haven't heard a lot about that, but I'm going to talk quite a bit about oxidative stress and how that impacts inflammatory responses. I'm going to talk a little bit about SNPs. I don't have a ton of slides about SNPs in here. That is one of my passions. But I will talk about that. And then how the complexity of what we're eating affects our genes. Oh, sorry. They're single nucleotide polymorphisms. So that's where you'll see SNPs. So those are just small variations within a gene. So the two most, this is kind of a little bit of a reminder, more basic information. This is when you're talking to patients. So patients come in and they're talking to you about inflammation, and you hear that a lot. You don't hear patients talking about oxidative stress and antioxidants. And antioxidants were all the rage a few years back, and you don't hear a lot about them now. But I want to point out how important they are, because they're the complexity of inflammation and oxidative stress, they go hand in hand. So inflammatory cells will release large amounts of what are called reactive oxygen species. So these are those ROS. What happens in chronic inflammation is there's an excessive amount of reactive oxygen species that are released, and then this initiates that cascade of inflammatory signaling. And that's where you see this upregulation of genes that code for these pro-inflammatory mediators. So things like TNF-alpha and IL-6. The paradox here, though, is, of course, nature is complex. Our human bodies are complex. Our food is really complex. But these reactive oxygen species, these ROS that we would kind of like liken as, you know, the bad guys if you're talking to a patient, we actually need these to kill microorganisms. So for apoptosis of damaged cells, what's used in that case are these reactive oxygen species. And it's kind of interesting because there was a hypothesis that was proposed by the Nobel laureate Watson back in 2014, and he was suggesting that chronic disease is actually caused by a failure to generate sufficient reactive oxygen species to get rid of some of these damaged cells. So it's kind of interesting, again, adding to the complexity, which almost every slide I will point out that this whole concept of inflammation and epigenetics is really, really complex. And so we can make somewhat general statements, but there's always this subset of compounds that have different actions that don't seem to make sense. But as we learn more and more, then it starts to make sense. So our food is medicine. We say this every day to our patients. What we learn from science is that our food, again, is very complex, has a ton of phytonutrients in there. So when you're talking to your patients about nutrition, instead of just talking about the combination of, the percent fat you should be on, talking about keto diets, what needs to really be emphasized is the types of plants that are in their diet. And diversity, I'll talk a little bit more about that, but diversity, it turns out, is extremely important. So it may be probably more advantageous, it looks like, to consume a little less plant compound. So I often describe this to patients. So if you were going to eat 5 cups of broccoli a day, and we know broccoli is like a superfood vegetable, if you're going to eat 5 cups, you would probably be better off eating 3.5 cups of 15 different plants. So that complexity is extremely important. So even when you're talking to patients about putting together meal plans, you want to emphasize that vegetable portion of your plate, which we're often pointing out should be half of your plate. That vegetable portion needs to be a mix of vegetables, and they need to be herbs and spices. So you're getting this whole array of plant compounds. And what they do, they're protecting our DNA. It protects it from oxidative stress. So there are direct antioxidant effects from some of those compounds that are found in the plants. And then there are these indirect antioxidant effects. So that's where you're enhancing the endogenous antioxidant systems. You're upregulating enzymes and natural antioxidant systems. So NF-kappa B is, as Bob pointed out, that turn-on switch for inflammation. Again, another complex chemical. It regulates genetic expression of inflammatory cytokines and enzymes and microRNA, and I'm going to talk about microRNA towards the end of this presentation. So hydrogen peroxide will activate NF-kappa B, and antioxidants are blocking NF-kappa B. That's kind of a very simplistic way to look at it. But so we think, NF-kappa B, whatever we can do to block that, but it turns out that NF-kappa B subunits actually contribute to these gene clusters that are necessary to resolve inflammation and relieve oxidative stress. So it is important, I think a couple of speakers have pointed out that inhibition is not necessarily exactly where you want to be. It's this concept of down regulation and like complete inhibition, which is done often with pharmaceuticals. This is where the beauty of plants comes in terms of down regulation instead of inhibition. So another big player is something called NRF2. And many phytochemicals have been shown to increase the activity of this nuclear factor derived protein. NRF2 is really important in modulating the antioxidant response. So you'll even see compounds in the marketplace that are designed as NRF2 activators. So this is kind of like your good guy. If NF-kappa B is what you're trying to address in terms of downregulation, NRF2 is what you'd want to upregulate. So now we start to look at plants. Where do we see some of these bioactives, these phytochemicals in plants that modulate the endogenous antioxidant network via that NRF2 pathway? So beetroot has, and these studies will be done on individual ingredients, right? So, and I do have some reference to comparisons between individual ingredients and then the whole plant extract. Curcumin and turmeric, another potent activator of NRF2. Sulforaphane and broccoli, so that's been getting a lot of press in the last couple of years. EGCG and green tea. Green tea is always something that comes up in many different places related to inflammation and oxidative stress. Resveratrol, which you're going to find in red wine and grapes. And then even ashwagandha extract has the ability to manipulate that NRF2 pathway. Quercetin, which we know is in high amounts in onions and garlic, but also is found in buckwheat and elderberry, if you can find any elderberry in your network. I don't know, in our community, I've had patients, and even like within our Facebook town community, elderberry is like a hot ticket item this year. Like everyone knows about elderberry, and they're online saying, get elderberry because it'll save your life from the flu. And so the shelves are cleaned out. It's kind of interesting. Beet leaf phenolics. So this slide is a comparison of the different parts of the plant. So Swiss chard has that bright red leaf. It's in the same family as beets. And that distinctive red color is due to a polyphenol called beta-cyanin. And it's a little bit different chemically than anthocyanin. And it also contains other phenolic compounds. So this total phenolic content is correlated to antioxidant activity. So you can see that red leaf, which has a higher content of beta-cyanin, but also has other compounds that are probably producing this synergistic effect, has the ability to scavenge those free radicals. And then the white leaf also, so this is where these other compounds come in. Even the white leaf chards have the ability to do this free radical scavenging, and not as much, it turns out, in the stems for this particular case. Now, when it comes, and I'm not saying don't eat the stems, because I'm sure, like, this is just one aspect of what it does. And we know in many plants, you're better off eating the whole plant because of the complex nature of our food. And here's our big problem, is there's a vegetable deficiency in the United States. So when you start to look at different categories, and especially this slide even singles out women and different age groups. Basically, if you look at it as a whole, 90% of the US population doesn't meet the recommended daily vegetable intake. And so the green lines there are the percentage of people who are falling below just the basic recommendations for fruits and vegetables. Or sorry, this slide's just vegetables. And then the red is meeting recommendations. So you can see that the 50 to 70-year-olds are doing a little bit better on their vegetable intake than pretty much every other class there. And you would think that the kids would be doing tremendous because, I've had this discussion lately that people tend to change their diet when they start to have children. They're concerned about what their children are eating, but those are probably more small subsets that you're seeing in your clinical practice and not necessarily representative of the entire population. Things could be changing. This is a slide from 2015. But look at that little black part where people are eating above the recommendation. It's kind of sad. So this is why you're out there, you're in the trenches, you have to explain to people why it's so important to eat vegetables. And sometimes they're overwhelmed, like how many get that many servings of vegetables in? 5 servings of vegetables? And those are like a half cup serving, basically. It matters, only 1 serving matters. So we know that and this is a meta-analysis of 17 cohort studies, that higher consumption of fruits and vegetables decrease risk of all-cause mortality. And 5% reduction for each serving of fruits or vegetables, which is, pretty mind-blowing that that goes up to 5 servings, and then you can see it kind of levels out there. So when you're working with a child who's basically eating no vegetables, you start to introduce, my approach to that is that I start to introduce it a little bit slowly because radical change is really difficult. And I even do this with my older patients, that I start to split like their after-school snack, for example, instead of just their goldfish that they have or whatever crackers they're having after school. Then I'll have the parent give them like carrots, half an apple. They have to actually eat a serving of fruit or vegetable, and then they can have their goldfish, but half of that serving. So this is how you start to transition kids, because it matters, like even a little bit matters. Then there's the concept of plant synergy. So any of you who have had a lot of herbal medicine training, there's this traditional concept of plant synergy, this theory that the combined effect is more than the sum of the equal parts. So if you look back to herbalist training, botanical medicines, there's always an emphasis on using the whole plant with some standardized ingredient in it, but still, so you can have a standardized amount of a particular compound, but still have all those different ingredients in there. And that's the beauty of the whole plant, because it's so complex. All those compounds are there for a reason, to provide balance. And then, as I pointed out, diversity is still really important, so you need to have a lot of different compounds in your diet. And this also comes into play. I'm not going to talk much about the microbiome, but that plant diversity is really important when it comes to the microbiome and the genes related to the metabolome. So this is a nice slide looking at the different colors in plants and where some of these compounds come from. So again, emphasizing the whole is greater than the parts. So if you start to look at the green parts of plants, that's where you're going to see compounds like isothiocyanate, EGCG, the isoflavones, lutein and zeaxanthin. The red parts, you're going to see things like ellagic acid and lycopene. White still, as I showed you on that slide with the leaves from the chard, you're going to see compounds like allicin, quercetin in there, and then in the purple and blue range, anthocyanidins, resveratrol. We know that anthocyanidins are really important even for cognitive health. There's a study showing children who consume blueberry extract, like freeze-dried blueberry extract, that they studied their academic performance on a number of different tasks within hours of consuming that extract, and it was enhanced. So think about that. Any of you have kids? My kids have blueberries every morning, hoping that it all matters. That can contribute to how well they're doing in school that day. Yellow and orange compounds, beta-carotene, alpha-carotene, and hesperidin. All really important compounds. And so getting them all together, that's where you're going to see that plant synergy. Here's a great example of the difference between just one single component, rutin, and the whole buckwheat extract. And what we see up until 20 micrograms per milliliter, you're seeing that the whole extract outperforms rutin until you get above that level. So again, antioxidant activity enhanced by other compounds in the plant. Other foods, we know that we've seen such evidence of this plant synergy. So the whole tomato fruit is shown to be stronger in terms of the benefits achieved than just lycopene alone. And we know that tomatoes are going to also have carotenoids, and they're going to have polyphenols. So even when a patient comes to me with concerns of prostate health, one of the things that I prescribe to them is tomato paste. So they may be on supplementation as well, various very targeted supplements, but I always incorporate tomato paste into the mix exactly because of this reason. Broccoli and Brussels sprouts upregulate phase 2 enzymes greater than those isolated glucosinolate metabolites. And then think about food combining as well. Broccoli and tomatoes are more potent when consumed together than alone. And we also know this is true even for absorption purposes when you start to look at olive oil and tomatoes together. Cranberry polyphenol mixtures have been shown to have synergistic effects. And then when you start to look at specific compounds, you can see that these studies are really easy to do, where they take individual ingredients and combine them, and they see things like quercetin and ellagic acid showing reduced proliferation above and beyond what the combination would do on their own. These are some interesting studies and good examples that I often use with my patients about why it's so important to eat all these kind of superfoods in their diet daily. So chocolate, and I've met, I don't know, under 10 people who hate chocolate, like hate and despise chocolate, and it always surprises me. But chocolate we know is an epigenetic modifier. So there was actually a study done in Spain where people were fed chocolate, they were force-fed chocolate, and then their global methylation status in their white blood cells was measured, and it expressed this favorable pattern of hypomethylation. So when you start to think, methylation patterns vary depending on the cell type, whether the cell is healthy or unhealthy, but in this case, hypomethylation, which is that epigenetic change, is the favorable profile there. Now, interestingly, people in this study who had the MTHFR and MTRR SNPs, those polymorphisms, didn't display as pronounced an effect. So this is where, and I'm not going to focus much on this today, but genetic testing is valuable, so you know why you have, and this kind of helps to explain why you give some patients a protocol that you think would work for someone else, and you have these great responders, and then you have the group that responded okay, and then you have a group that responded poorly, or even the reverse effect of what you expected. And this all has to do with our genetic differences. And even the differences, when I say genetic, I'm talking about our human genes, but then the genes of our microbiome. Green tea can also have an effect on methylation. So hypermethylated DNA, in this case, that would be an adverse methylation pattern, can undo. So EGCG can actually undo that methylation. And what we know is that it can actually affect tumor suppressor genes. So it can upregulate these tumor suppressor genes, and that's basically where this anti-cancer effect comes in. Then we have coffee, another superfood. So tea and coffee. And tea is the second most drank beverage in the world, next to water. Coffee contains caffeic and chlorogenic acid, and that inhibits DNA methylation as well. And then, of course, turmeric, a potent hypomethylation agent. So all kind of on my master list of superfoods, and cost-effective, easy to get into your diet, all important epigenetic modifiers. Okay, and microRNAs. So you're hearing a lot about microRNAs today. A really fascinating, rapidly evolving field. We don't know, we know a fraction of what there is to know. I guess we don't even know what we need to know about it. But basically, there's a new division of science, just like we had genomics, epigenetics. Nutrimyromics is the study of the influence of diet. So we had nutrigenomics, now we have nutrimyromics, the study of the influence of diet on modification of gene expression due to the epigenetic processes related to these microRNAs. So these microRNAs, I'm going to keep repeating what you've been hearing all morning, they're small, non-coding RNA molecules, 18 to 25 in length, and they exert effects on post-transcriptional gene silencing. So they're down-regulating for the most part. Again, complex, so some of them upregulate. But one microRNA has the ability to affect hundreds of messenger RNAs. These are controlled by the environment. So of course, when we look at, I'm talking about food today, and we know the same carries for microRNAs. Likely that carries for epigenetics as well, meaning that stress has an impact on expression. And then exercise, mindset, we all know this affects DNA expression. But I'm going to focus on diet. So bioactives in food have been shown to impact endogenous expression of microRNAs. So phytochemicals, again, your big players, and this is just from the research so far. And again, this is a field that is expanding, but there are a limited number of studies. Resveratrol is definitely been shown to affect microRNAs. Curcumin, quercetin, and then many of the proanthocyanidins from green tea, from grapeseed, from chocolate. So again, when we look at foods like a complex food like chocolate, a complex food like coffee even, there's what we thought we knew, we don't know what we don't know. Same carries through here. Even vitamins affect microRNA production. So vitamin D is really important here, vitamin A, and then some of our minerals like that we know are extremely important for inflammation and oxidative stress, selenium, zinc are really important, and then even certain fats have been shown to affect microRNA expression, saturated fatty acids, and polyunsaturated fatty acids. So these research studies, and this is just a small snippet, you can find large-scale lists of what each ingredient, which microRNA is affected, what pathway is affected in that has to do with oxidative stress and inflammation. So we have this endogenous microRNA. So what's interesting is that there's also exogenous microRNAs. There are microRNAs that are actually found in our food. So you can actually measure microRNAs in the food. So now I feel like when I learned this opens up a whole new body of information when you think about why is it important to eat whole foods, right? Like you can't just give your patients supplements and they eat no vegetables. they're not going to heal. They're not going to get better. So is part of what's going on the microRNAs that are found in our food? We actually find those in the intestine. They can actually survive digestion and get into your stomach. Measurement of these microRNAs in serum is mixed. So some of the studies show, and this may be just issues with measurement techniques, but microRNAs from plants tend to not be at almost undetectable measurable amounts in serum, yet microRNAs from like dairy and animal compounds are at higher levels. But again, this might have something to do with measurement techniques. Again, it's an evolving field. There are also studies that look at microRNAs as a potential biomarker for certain disease states. So you can actually look at biomarkers in even saliva and urine as a potential biomarker. There's one paper about microRNAs as a biomarker for stress as a salivary test. So that's really interesting. So for us as clinicians to start to see, like, there are tests we don't even know that could potentially exist that we'll be able to look at. And then you can measure a lot of this in the intestine, and the question is, is there some other mechanism going on? So the same thing with quercetin. A lot of you know quercetin is not, and curcumin is not absorbed at a high rate, that it hangs around the gut, but there are a lot of different things that it does in the gut, and there's a purpose to the effect that it's having in the gut. Perhaps the microRNA has some similar mechanism as well, that it doesn't necessarily require high absorption level, but we don't know. And I'm just going to end with more complexity about inflammation, oxidative stress, and SNPs. So some of you, a lot of you probably know a lot about MTHFR, MTRR, some of the methylation SNPs. Those are important, but there are actually a whole subsets of SNPs that have to do with inflammation and oxidative stress. There are genomic panels that you can do to test for some of these. Certainly if patients have problems with detoxification and methylation, it plays into all of this. But the inflammation SNPs, there's IL-6, there's NCF4, and then there are all these CpG SNPs that have to do with cardiovascular disease. Then you have a whole array of antioxidant enzyme SNPs. The manganese superoxide dismutase is a really important one. Even a SNP for catalase, which protects against reactive oxygen species. And then glutathione transferase is also quite important. So this just is another layer in the complexity of dealing with people and nutrition, and why, the more digging that you have to do to figure out what's going on with them and where the dysfunction is and how you can address that through nutritional therapy. So it all goes back to our food, right? So I'll just end with that slide, and thank you.